The purpose of the article, Sex-Linked Diseases, the case of Duchenne muscular dystrophy, also known as DMD, is to determine why the disease allele frequency has not gone down even after the prenatal diagnostic testing that now determines whether fetus carry the debilitating or fatal sex-linked mutation. The American biologist known for his insight into the genetics of X-linked inheritance, among many other things, is Thomas Hunt Morgan during the early 20th century. Although X-linked inheritance has made for a fascinating area of research for the likes of Morgan and many other biologists, this topic creates space for interesting questions about the societal impact of reproductive technology. This is because many defective X-linked mutations in humans, particularly recessive mutations, are associated with debilitating and sometimes fatal diseases. Although DMD is named after the French neurologist Duchenne did first describe the disorder in the late 1800s, the cause of the disease remained a medical mystery until the mid-1980s, at which time the DMD gene defect was identified. Now, major findings throughout the article include that DMD is a fatal neuromuscular disorder caused by a recessive mutation on the X chromosome. Because the related mutation is recessive, DMD is more common in boys than in girls, as boys do not have another copy of the X chromosome to compensate for the genetic defect. DMD affects about 1 in 3,500 males. On the other hand, most girls born with DMD mutations are merely carriers because they each possess only one mutated DMD gene on one of their two X chromosomes. Although not themselves affected by DMD, carriers can pass DMD genes onto their children. It is now known that the DMD gene encodes dystrophin, a huge muscle protein with about 4,000 amino acids that plays an integral role in maintaining the structural integrity of muscle cells. Cells with mutated DMD genes cannot make normal dystrophin, which means that they are unable to function appropriately. Scientists have been researching for ways to cure DMD for as long as they have known about the disease. Today, based on the wealth of knowledge about the underlying genetic and molecular mechanisms of DMD, researchers are actively testing a variety of different therapeutic approaches. Given the information in this article, I found the fact that prenatal testing upsets the balance of these mutations that are ousted from the gene pool by natural selection very surprising. As humans in today's world, one would think that testing for a fatal mutation beforehand would be a very great help of scientific technology. However, prenatal diagnostic testing and embryo sexing for sex-linked recessive disease mutations bring up more than ethical issues. They also raise some interesting questions about how we as society can affect the genetic structure of the human population at large, as Hastings, 2001, emphasized in his study. Normally, a balance exists between mutation and selection. Sex-linked disease genes appear over time because affected individuals often die before they reach reproductive age or are unable to reproduce. This leads to why Hastings believed that the prenatal testing disturbs the natural balance. Out of the 3 reference articles given, I chose Reproductive Technologies, a Long-Term Cause because of the simple fact that its title lured me in, leading me to wanting to know more. This article mainly highlights how the human society is far from ideal, which should come as no surprise, but still such a thought has rarely troubled population geneticists, though as their theoretical models are based on the behavior of ideal populations.